All right, then. I will now begin the session. My name is Keegan Brown. I'm a part of the Public Health Alliance for Genomic Epidemiology. Today, we will be having a session regarding the introduction to public health phylogenetics. Our speaker today is Dr. Zoe A. Dyson from the London School of Hygiene and Tropical Medicine. The program layout will be as follows. I'll have a quick 5-minute introduction to Zoe's background. I'll then hand over to Dr. Dyson in order to present for us, after which we will be having a Q&A session for about 10 minutes, and then we will have the last 5 minutes to just close off the session. Please keep all your questions till after the presentation, or feel free to post them in the chat. Thank you very much. All right. It is my pleasure to introduce today's speaker, Dr. Zoe Dyson, an assistant professor of bacterial pathogen genomics at the London School of Hygiene and Tropical Medicine. Dr. Dyson is an expert in genetic epidemiology and the evolution of antimicrobial resistance in enteric fever-causing bacteria, especially Salmonella typhi and Paratyphi A. She holds B.Comp/B AppSci honors in computer science and biotechnology, along with a PhD in microbiology. Additionally, Dr. Dyson is an associate fellow at the Higher Education Academy and visiting researcher at the Wellcome Sanger Institute. Her research is critical to understanding the antimicrobial resistance and improving treatment strategies. As part of PHAGE's ongoing work at the intersection of phylogenetics and public health, we've launched a phylogenetics working group led by Rita and Shavari to develop a guidance document on best practices within this field. We're excited to offer a series of training to support this effort and encourage collaboration. Please join me in welcoming Dr. Zoe Dyson. Over to you, Zoe.
Thank you, Keegan, for the lovely introduction, and also thank you very much to PHAGE for having me here today. It's a pleasure to speak with you all. Could I just please confirm that my slides are appearing?
Yeah, we can see them.
Great, thank you very much. So hello, everyone. As Keegan said, my name is Zoe Dyson. I'm an assistant professor at the London School of Hygiene and Tropical Medicine. And today I'm going to be walking you through an introduction to public health phylogenomics. So today in this webinar tutorial, we're going to be covering what are phylogenomics and genetic epidemiology. We're going to walk through how to read and interpret a phylogenetic tree, and we're going to bring our understanding together in a case study that combines metadata, typing data, and phylogenomics to understand AMR transmission and transmission dynamics of the pathogen more broadly. So firstly, let's talk about what are phylogenomics and genetic and genomic epidemiology. So we can broadly think about phylogenomics as the study of genetic relationships between different biological entities. And I'm saying biological entities here to account for some scientists sort of firmly believing that viruses don't count as organisms, simply because they're not considered truly alive in some ways. So I tend to use this catch-all term when having these discussions. And this idea of phylogenomics isn't a particularly new idea. It was first proposed by Charles Darwin in around 1837. And you can see in his notebook capture here that he's outlined this concept of how we could consider describing particular biological entities and how they might be related to one another. So in molecular phylogenetics, this is whereby we use molecular data from biological entities. Maybe we're looking at nucleotide sequences of individual genes or amino acid sequences of proteins to infer evolutionary relationships of different biological entities. And for this webinar, we're largely going to focus on using whole genome sequencing data, that is core genome nucleotide alignments of bacterial pathogen genome data to infer phylogenetic trees. Now, there will be some overlap with other organisms, particularly when it comes to reading and interpreting phylogenetic trees, but I'd just like to keep in mind that that is the focus of how we're framing today's session. So the question is, well, why would you want to use whole genome sequencing data? And I think as many of us are becoming increasingly aware, genomics is becoming a core method for pathogen characterizations in both research and public health laboratory contexts. And there's several reasons for this, but I think the main ones that come through is that it's a robust method, it is reproducible, and it's informative, because we know that genome data are enriched for many different aspects of pathogen biology. We can extract information on drug resistance, pathogen virulence, and different types of lineages circulating in given settings at different scales. But also we can use this genomic technology to understand pathogen transmission dynamics at different scales. And for me, I think one of the key benefits of working with genome data is that they're readily stored. I know we all have difficulties with storage in any given setting that we're in, but broadly speaking, we do have large global databases where we can deposit and access genome data, we can share them with colleagues, and we can reinterpret and reanalyze genomic data as our understanding of pathogen biology continues to develop. For example, as we understand new molecular mechanisms like genes or mutations that are associated with drug resistance, we can always go back to data and we can actually reanalyze and reinterpret data to understand best when, where, and how, for example, resistance to a given drug arose and has been spreading throughout pathogen populations. And as I've been alluding to, there's a lot of different information we can extract from pathogen whole genome sequencing data. We can look at the molecular determinants and mechanisms driving antimicrobial resistance, and sometimes with sufficient sampling, we can actually look at AMR prevalence on a much greater scale. For some organisms like Klebsiella pneumoniae, there are specific virulence factors that are important for community infections in healthy individuals. For example, different siderophores that are iron scavenging molecules are important to this, and certain capsule types that confer a hypercapsule state that are associated with enhanced virulence as well. And we can extract information on those using whole genome sequencing data, and looking at surface antigens like capsules can be really informative for how we think about targeting different pathogen populations circulating in different regions using vaccination strategies, and it can help guide some of those. And as I mentioned before, we can extract information on the different lineages or strain types of pathogens and how these are spreading on different levels. We can look at spread across different patient cohorts, across different geographies, across regions, and globally as well. And these are just a few examples of the types of information we can extract from whole genome sequencing data. I could literally talk all day on this one particular aspect of why I think genome data, in particular whole genome sequencing data, are really excellent data to work with. So for those who may not be familiar with strain typing techniques, I just wanted to give one example of one that's relevant to the case study later in this presentation, and that's our genotyphy Salmonella typhi genotyping tool. So what I'm showing you here is a large phylogenetic tree that contains around 2,000 whole genome sequences of Salmonella typhi. And as those of you who've worked in phylogenomics for quite a while, and those who might be new to it can probably estimate, inferring large contextual trees like this every time you want to understand the lineages circulating in a pathogen population can be quite a lot of work, and it's not always needed depending on the type of analysis you want to do. So by using this large phylogenetic tree, we developed a backbone understanding of the different lineages that were present in the pathogen population structure, and we're able to develop this genotyping scheme that could rapidly identify key mutations associated with branches in the tree to tell us at a broad sort of level what particular lineages we were looking at in new data as they came in. So for example, if I see genotype 431, I know that it belongs to this cluster here down the bottom, and I immediately know, based on data I've seen before, that this is a global genotype that's associated with intercontinental transmission and antimicrobial resistance. And there's a variety of different typing schemes out there. There's a similar scheme to this for both TB and Shigella sonnei, and we also have multi-locus sequence typing as a broader approach to this as well. So we've talked a lot about why genome data. I guess the next thing is, well, why phylogenomics? So there's a variety of different ways that phylogenomics can enrich our study of bacterial pathogens. We can use it to characterize newly emerging threats when we start to find variants that may be more difficultly controlled coming through in diagnostics. So we can understand how these have emerged, what they're most similar to, and start thinking about control and intervention strategies. We can use phylogenomics to understand pathogen transmission dynamics, and this can be particularly helpful in outbreak scenarios. And this can be done between different patient cohorts across different cities, regions, and different countries. And when we have a large sort of bank of data, this can sometimes help us do pathogen surveillance at a global level as well. If we understand which locations have high burden of disease and we understand what serotypes are circulating there, this can ultimately help guide control and intervention strategies like the design of effective vaccines and opportunities to further drug development. And again, there's many different uses for phylogenomics that you might have, but this is just to outline a few potential opportunities that are mainly related to the pathogen space. So for the rest of this tutorial and webinar, we're going to be focusing on transmission and outbreak investigations and understanding transmission dynamics. Which brings me to pathogen genomic epidemiology. So as some of us might be aware, epidemiology is the study of the distribution, determinants, and causes of disease to help us prevent and control them more effectively. And I've just highlighted here a classic example of epidemiology. This is John Snow's map of the London cholera epidemic from 1854, where you can see that he's outlined in points different cases on this particular map, sort of in and around the Broad Street location here. And this was a case where there was a large outbreak of cholera occurring in London, and John Snow set about investigating it. And for the sort of the methodology he's applied here, he's considered a founder of modern epidemiology. He was able to identify in the end that there was a water pump on Broad Street that was disseminating contaminated water that was feeding this cholera outbreak. And when they removed the handle from the pump, the epidemic did then subsequently wane. So when we think about molecular epidemiology, this is about using molecular data to inform the goals of an epidemiology study. And for the purpose of today's webinar, we're going to be talking about using pathogen genomic epidemiology. This is the use of pathogen genomics to understand infectious disease epidemiology. Which brings me to an important point, is that genome data alone, while they're really interesting and really exciting, and I certainly love looking through a new data set when I'm able to, having the genomes alone usually isn't enough to be informative to understand pathogen spread and transmission dynamics. Genetic epidemiology or genomic epidemiology is about adding other data to genomes, and usually phylogenomics as well, to learn more about the epidemiology of a disease. So, for example, if we were to include clinical information and patient data, for example, we could include age or sex here to understand if transmission is occurring more intensely among particular age groups that might suggest we should target, for example, an intervention like a vaccine towards specific age groups. That is something we could unlock by including additional data. Similarly, if we noted that there was enhanced transmission among males or females, we could target specific interventions to those particular groups. And by including spatial and temporal data, such as country of isolation, ward of isolation, or date of isolation, we can start to think about understanding geographic origin and spread of a particular pathogen, as well as potentially reconstructing the transmission chain. And there are other opportunities in including, say, laboratory antimicrobial susceptibility data, as well as genome-derived data on antimicrobial mutations or genes, as these alignment of these data against a pathogen population could help us identify where we are missing information on how resistance is occurring in a particular population. So we've talked a little bit about why we want to use genome data, why we might like to use phylogenomics, and what we mean when we talk about genomic epidemiology or genetic epidemiology. I'd next like to go through how to read and interpret a phylogenetic tree. So this will probably be familiar to some viewers on this particular tutorial webinar, but it might be new to others as well. So we're going to start from the basics. So what is a phylogenetic tree? Well, it's a really central concept to phylogenomics. And there's a lot of different ways that you can infer a phylogenetic tree. There's all sorts of methods available to do this. And accordingly, lots of different software implementations of those algorithms. We have maximum parsimony, neighbor joining, maximum likelihood, and there's also Bayesian methods for creating dated phylogenies. But broadly speaking, phylogenetic trees, like the one that I've indicated on this slide, are graphs that show you genetic relationships between different sampled taxa leaves, tips, or external nodes. And what these tips, external nodes, so on and so forth, leaves on a tree indicate are the sequences that have been provided to the algorithm in order to infer evolutionary and genetic relationships between the organisms that you wish to study. And when we look at these graphs, we know that A, B, C, D, and E are the sampled taxa or the sequences we've provided, and that these external nodes are led to by branches in the phylogenetic tree. And these represent horizontal edges that usually indicate genetic distances. So, for example, in a maximum likelihood phylogenetic tree, these might represent substitutions per variable site in the alignment that you've provided to the software to then go ahead and infer the evolutionary history and genetic relationships between these particular samples. However, if you're looking at a dated Bayesian inferred phylogenetic tree, your branch lengths might also represent time. So when you're looking at these in the literature and other studies, it is quite important to understand what methods have been used to infer the tree that you're looking at and what the different portions of the graph might mean in terms of the branch lengths and how you interpret those. Next, we have internal nodes of the tree. So for example, if we look at sequences A and B here, we can see that these tips, when we move backwards on the branches, are connected using vertical lines that just show the connections here, and that they lead to this internal node here labeled X. And these are effectively the ancestors of sequences A and B that have been inferred by our phylogenetics inference software. And we don't know the sequences of those particular inferred ancestors. If we want another example here, we can see that internal node Y has several descendants. It has C, D, and E, but also another ancestral or internal node here, W. Now, if we move all the way back through the branches in the phylogenetic tree, we come to this node here called Z, and this is sort of a special node as this is the most recent common ancestor of all of the nodes and sequence studied in this particular tree, and it might also be referred to as the most recent common ancestor or root node. So phylogenetic trees can be rooted in a variety of different ways, and they can also be unrooted. So if we were to review an unrooted tree, for example, I just provided another toy example of this here. If we were to look at this unrooted tree, what it can tell us about is the relatedness of the different taxa or sequences that we're studying without making any particular assumptions about ancestry. But if we wanted to take this unrooted tree and root it, we could do so using a midpoint method. So when you select this option in the software, what it's really doing is it's looking at your unrooted tree, it's finding the longest path between any taxa in this given tree, and it's actually going to fold the tree in half, halfway along that particular path through the tree to give you a midpoint root that usually represents the most recent common ancestor in the particular tree. It approximates this. So before we carry out that rooting, we can see that from this example here of the unrooted tree, we can see that A and B are more closely related to each other than C or D. But when we apply this midpoint root and get this resultant tree here, what we can see is we then have some directionality to the evolution in that particular tree. So midpoint rooting is one option. Another option that's available to you in rooting phylogenetic trees is what's referred to as outgroup rooting. So in this scenario, what you do is you include your in-group taxa that you want to study in your inference. So in this case, this is sequences B, C, and D. And you also include a more distantly related taxa. So for example, A here is something that is slightly distantly related that gives a reference point for the evolution and the direction of evolution represented in this particular tree and helps us find the most recent common ancestor. So, for example, if I was inferring a phylogenetic tree of a lot of different Salmonella typhi lineages, I might consider rooting my tree with a paratyphi A, sort of a more distantly related serovar of Salmonella enterica, but I probably wouldn't go so far as to root this tree with an E. coli because that's quite a distant evolutionary relationship there. Now, I think an important thing to know here is that you don't have to just use one outgroup. For example, you can choose to use a selection of different outgroups, and I'll show you an example where we've done that in a case study. But if we go back to this example of Salmonella typhi, I'm looking at a lot of different genotypes or lineages. I could also use a paratyphi A, a typhi Miriam, and an Iteritidis as sort of more distantly related serovars of Salmonella enterica, subspecies enterica, to provide that reference point for the evolution shown in the tree. So I think it's also important to note that while many people, when they carry out phylogenetic or phylogenomic analyses, do use an outgroup route, they would generally prune this branch out and not actually display it in the figure in the manuscript. And the reason for that is that by including this sort of more distant, longer branch that appears, it can often compress the data that you really want to show when you really want people to understand the relationships between. So a lot of the time it'll be pruned out, but sometimes you might see these sort of slashes occurring across the branch leading to the in-group to indicate that there is an outgroup and what that outgroup is. So there's those two main visualization options that people use. So we talked about the basics of reading and interpreting a phylogenetic tree. I'd also just like to spend a moment talking about the language we can use when we want to refer to portions of the tree. So, for example, if I was referring to a clade or a lineage, what I'd really be referring to is a section of the tree consisting of an internal node, so for example, Y here, and all of its descendants, W, C, D, and E. Now, if I had two clades that I wanted to refer to that shared an ancestor, sometimes I might choose to refer to those as sister clades, and we'll talk a bit more about that in the case study later on. And this term is used a little more rarely than the others, but sometimes if you have two adjacent tips sharing an ancestor, so A and B sequences here share an ancestor X, you might refer to those as cherries because they do look a little bit like the characteristic picture of a cherry of two cherries on a stem. Now, there are some more sort of terms that describe specific evolutionary relationships that you might also want to use. So, for example, if I had screened all of these isolates A, B, C, D, and E for antimicrobial resistance determinants, and I found that these three isolates, C, D, and E, shared an antimicrobial resistance gene, if I wanted to talk about just that clade, I might say that it's a monophyletic group. So it's a group of an ancestor and all of its descendants, so Y, W, C, D, and E. If I wanted to talk about a portion of those data, so I wanted to talk about C and D and W and Y, but I was leaving off E, I could refer to that as a paraphyletic group. So I saw, for example, that C and D had acquired an antimicrobial resistance gene that had been lost in E. I could highlight this as a paraphyletic group that is carrying that particular antimicrobial resistance gene. And it's a way of referring to an ancestor, but not all of the descendants. I could also refer to a group like this as a polyphyletic group, and this is where we include the descendants but not all of the ancestors. For example, we're missing, say, X and Z that would be, X that would be involved with this grouping here. So an example of where you might refer to a particular group of strains or clusters as being polyphyletic would be when we look at the evolution of Shigella in the broader E. coli tree, because we have several points of convergent evolution where Shigella has picked up a virulence plasmid, but we wouldn't be talking about all of the ancestors because that's a specific event that's occurred in several different clades. And for some phylogenetic trees, you may have the option of inferring bootstrap support values. So I've just got this example of, again, a very small tree here where I've annotated numbers at the internal nodes of the tree representing these bootstrap support values. So in this example, I'm showing these as out of 100, but you might also see these represented as a proportion, say 100 might be represented as 1, 95 might be represented as 0.95, and so on and so forth. And the benefit of including bootstrap support values is that these allow us to understand the confidence that we can have in various parts of the inferred tree's topology. And the way that this works in most software implementations is that the software will carry out random subsampling of the alignment that is used to infer the tree that contains information on sequences A, C, B, and D. And it carries out random subsampling by site with replacement and reruns the trees. And this is usually done 100 or 1,000 times and is often referred to as pseudo-replicates. So when you're looking at these bootstrap support values, what they're really giving you is a measure of how many times the same clades or clusters appeared in the individual tree. So, for example, if we were to look at A and C here, we can see that they probably were clustered together 100% of the time, whereas this broader cluster occurred 95% of the time. So we've talked about phylogenomics and genomic epidemiology. We've done some revision or some learning on how we can read and interpret a phylogenetic tree. I'd like to now bring this understanding together in a case study that combines metadata, typing data, phylogenomics, to help us understand the emergence and spread of antimicrobial resistance, as well as pathogen transmission dynamics more broadly. So the case study I would like to look at today was a study carried out by Professor Sam Kariuki's team at the Kenyan Medical Research Institute, or KEMRI. So Sam and his team were interested in learning about causes of invasive salmonellosis in an informal settlement, Nakuru, in Kenya. And the study that Sam and his team carried out focused on children 16 years of age or less that were living in Nakuru. And the surveillance was carried out from August 2013 through to November 2016. So while this was a broader invasive salmonellosis study, a major focus of this work was Salmonella typhi, which, as some of you might be aware, is the bacterial agent of enteric or typhoidal fever. So for those who might not be so familiar with typhoid fever, what we're talking about is a fecal-orally transmitted infection. And we see around 10 million cases of typhoid fever annually, of which more than 100,000 result in mortalities. And due to this fecal-oral transmission through contaminated food or water, it tends to be a larger problem in mainly low to middle income country settings, where there's insufficiencies in sanitation and hygiene infrastructure controlling outbreaks of waterborne diseases like typhoid fever. So the mainstay of controlling typhoid for many years has been the use of antimicrobial chemotherapy. However, this is becoming increasingly more complicated by the emergence of antimicrobial resistant strains. And another complicating factor for the control of typhoid is asymptomatic carriage. So in a small proportion of individuals, around 3 to 5% of those infected by Salmonella typhi, the organism actually colonizes their gallbladder. So individuals will appear healthy, they won't have any symptoms of typhoid, but they will continue to shed the pathogen in their stool and subsequently contribute to onward transmission of the disease. And they can be quite difficult to identify in a community setting, simply given that they are asymptomatic. So what Sam and his team did was that they carried out surveillance of febrile individuals presenting at 4 healthcare providers in Nakuru, and this is a setting that's around 20 kilometres outside of Nairobi. And the individuals that were presenting as febrile in this study were defined as acute symptomatic infections or cases. At the same time, they carried out fecal sampling of healthy individuals attending healthy mother and child clinics. This was usually for nutritional advice or routine vaccinations. And these were healthy individuals that were still culture positive for typhoid, and these were asymptomatic carriers in the setting. So in total, Sam and his team sequenced 99 cases and 54 carriers or controls from Nakuru. And these were sequenced using Illumina high throughput sequencing platforms. So at the same time, Sam and his team were carrying out antimicrobial susceptibility testing on pathogens that were coming through from these given studies and these different patients. And they noted that there were high rates of multi-drug resistance, that is resistant to the classical first-line drugs used to control typhoid for many years. And this is chloramphenicol, cotrimoxazole, and ampicillin. And at the same time, they were seeing increasing rates of ciprofloxacin and non-susceptibility as well. So overall, what Sam and his team wanted to learn about is how antimicrobial resistant typhoid was spreading in Nakuru. So the first stage in this analysis was to learn about what pathogen genotypes or strain types were circulating in the setting. So we applied the genotypy genotyping framework I showed you earlier. And what Sam and his team found was that 95% of the isolates obtained during this study were one particular genotype known as 431. And for those who've worked with typhoid previously, they might recognize this under another name of H58, which was the previous typing scheme. So what we could see is that the next stage was to understand what we could learn through phylogenomics. So in this case, Sam and his team inferred a dated phylogenetic tree using BEAST with Nakuru data and also previously published whole genome sequence data that was available in the public domain from Kenya, which left 239 whole genome sequences to be analysed of Kenyan data. So with this dated phylogenomic approach, the branch lengths become calibrated using the date of isolation. So in this case, this was day, month, year, but in some cases, this might also be simply the year of isolation to calibrate the molecular clock to be able to convert branch lengths into years. So you note, instead of having a short scale with a number here, I've actually got a timeline down the bottom of this phylogenetic tree. And that's an example of knowing what type of tree you're looking at and knowing how to interpret the branch lengths that you see. And overall, from this analysis, what they found was that there was a substitution rate of 0.8 substitutions per genome per year, which is pretty close to 1 SNP in the genome sequence per year, which is consistent for typhoid. It is a slowly evolving pathogen. And by looking at the dates of these two major sublineages, 4311 and 43212, sublineages 1 and 2 of genotype 431, they could see that the pathogen emerged in this setting, these particular genotypes emerged in this setting from the late 80s to early 1990s. Now, the next thing was to include patient data in this phylogenetic tree. So you can see that, for example, the cases here have been coloured as red and the carrier samples have been coloured as blue in both the branches and this coloured bar next to it, just sort of to aid comprehension. And the previous data from other studies is coloured in grey. Now, what's really interesting here is if you look at the structure of the tree, what you can see is that pretty much all of the clades you can see in this tree are made up of carrier and case samples. And what this suggests is that they're genetically really, really similar, and that this intermingling is suggesting that you're seeing onward transmission of disease from both groups. And that, broadly speaking, what we can see is that cases and carriers are drawing their infections from the same pool of circulating pathogens in this particular setting. So this is consistent with contaminated water spreading the disease throughout the setting. So the next thing we wanted to do was Sam and his team really wanted to make sure that what we were looking at was convincingly carriers. So for this analysis, what they did is they identified that there were 3 different sublineages present in the tree here, 2 of this 4311 lineage 2 and 1 of this 4311 lineage 1. And what they then did was they looked at the terminal branches in the tree. And in this cartoon here, I've just sort of highlighted to you that when I'm referring to terminal branches, what I'm referring to is the branch that immediately leads out from our tips of the tree that represent our sequences. Now, terminal branch lengths are a little bit special, as you can consider these measures of evolutionary time since the isolate shared a common ancestor with another sampled isolate in the tree. And when we take these branches and we look at the measurements per individual sequence across these 3 lineages, what you can broadly see is that our carrier samples are more deeply branching. They have longer terminal branches. And when we directly compare those to our cases, that becomes quite apparent. And what this is suggesting to us is that we're seeing a longer duration between acquisition of the infection to sampling in the clinic, which would indeed be true of carriers who are healthy individuals and unlikely to be reporting to clinics where their samples would be picked up through passive surveillance and blood cultures. So we're pretty convinced that what we were looking at here was indeed true carriage samples. And we did some other analyses to look at selection pressures that were aligned with other data from cholecystectomy patients that are a little bit beyond the scope of this webinar, but we have reported in the full publication if people are interested in that. So we noted that we saw 3 different clades in that phylogenetic tree, and Sam and his team hypothesized that these might potentially represent 3 different introductions of the pathogen into Kenya and Mukuru as well. So what Sam and his team did is they inferred a maximum likelihood phylogenetic tree. So noting that with this particular type of tree, our branches represent substitutions per variable site. So we can think of them as genetic change rather than time that I showed in the previous tree. And a decision in inferring this type of tree in this particular scenario was really that with the techniques we had available at the time, i.e. the software beast, it gets quite difficult to process sample sizes larger than 300. So in this case, it was computationally more efficient and tractable to infer this maximum likelihood style of tree. So what we did was we inferred a global context tree that included around 1,200 samples, including the Mukuru data of genotype 431 isolates previously published and in the public domain. And what we did for this particular tree is that genotype 431, or H58 as some people know it, is a sort of fairly recently evolving clade. It's low diversity, so we wanted to be really certain of our tree topology and structure. So for this, we outgroup rooted the tree, but we actually used a set of non-H58 or 431 genotype isolates, indeed 61 of them, each representing each different genotype or clade of typhi that we're aware of, to get that rooting absolutely spot on. So next what we did is we overlaid study data and spatial data onto this particular tree. So you can see the study column is just showing you in these sort of dark blue flecks the data that came from Mukuru. And then if you look at this second column here, and indeed all of the branch colours in the tree, you can see that we've represented the location where all these genome data were isolated from. So where the data were locally collected, this is indeed the location they were collected from. In the case of travel-associated cases that are reported to public health laboratories in other countries, this was the location of travel where the individual picked up their infection. So we can see that in shades of blue and purple, we have data from Eastern Sub-Saharan Africa and South Africa. And then shades of sort of green, we tend to have our South Asian and West Asian data, and purple, we have Southeast Asian data. And locations that were more rare in our collection have been coloured black for other. So what we note here is that we do have 3 different monophyletic clades, all descending from a common ancestor, that are associated with typhoid populations that have spread throughout Eastern and Southern Africa. And when we look at the ancestors in each of these clades, coming back here, coming back here, coming back here, we can note that they're all green. And this is suggesting that we've seen 3 separate introductions of all of these, each of these clades into Kenya and other regions in Sub-Saharan Africa, and it can illuminate the transmission routes of those particular pathogens. Now, one thing that's quite interesting is that when you go back to this timed tree on the previous slide, is you can see that these 2 older introductions were occurring late 80s, early 90s, but there was also a more recent one down the bottom here shown in green. And we can see that in the tree, this indeed does split out into its own discrete clade of Eastern African strains. But what's interesting about this is that we see that we have 2 sister clades here. We have a dark blue colour of isolates here from Kenya and a lighter blue here from Uganda. And while we can see overall the ancestral sequences are indeed from Southern Asia, when it comes to the entry into Sub-Saharan Africa, because these are sisters, we know that there's been some connection in the transmission there, but we can't tell anything about the direction of that based on the tree structure. We don't have that information to fill in the gap. So it could be that it entered Uganda first before moving to Kenya, or vice versa. And Sam and his team hypothesized that there was a major road linking these 2 countries that could have acted as a transmission route. So the next thing we did was we overlaid our genomic typing data onto the phylogenetic tree. So this included our drug resistance data, our plasmid data, and again, some more clarity on the particular lineages that we'd inferred. So that was informative for understanding AMR. I'll talk more about that in a moment. But what we also did was, given that we had 3 distinct lineages that were present in Mukuru and Kenya, is that we actually wanted to define new genotypes to include those in the genotyphy genotyping scheme. So we identified point mutations representing the branch leading to this common ancestor for each of these, and they were subsequently designated East Africa 1, East Africa 2, and East Africa 3, and they could be rapidly identified through the typing data as well, and each of these representing a different transmission wave, including this newly noted one that hadn't been reported previously, East Africa 3, that is spread between Uganda and indeed Kenya. So when we overlaid our genomic typing data onto the phylogenetic tree, the common resistances we observed and the questions that Sam and his team had were about ciprofloxacin non-susceptibility that is driven by quinolone resistance determining region mutations in gyre A, gyre B, and PAR-C. This included multi-drug resistance, that is the acquisition of genes that reside on a composite transposon, conferring resistance to chloramphenicol, cotrimoxazole, and ampicillin, our classical first-line drugs for controlling typhoid. And also the plasmid commonly associated with those, which is known as an incompatibility group HI1 plasmid. So what becomes apparent is that ciprofloxacin non-susceptibility in our Makuru data highlighted here with these flecks of dark blue was really quite common, as was MDR, comprising 69 to 80% of isolates screened. And we zoom in on portions of this tree from the Makuru data, we can see that the evolution of ciprofloxacin non-susceptibility conferred by these single point mutations represented in green here is occurring in different clusters of the tree, indicating that there's ongoing emergence of ciprofloxacin non-susceptibility in this particular cluster here. Similarly, when we look at this newly emerging strain that's been picked up only in this study, this third transmission wave into Makuru and Kenya, what we can see is that we indeed have multi-drug resistance shown by these gold bars here, but we're lacking the plasmid. And this is suggesting to us that something that's quite common to typhoid has happened, is that the multi-drug resistance transposon has actually jumped from the plasmid, where it was originally identified, into the chromosome of the organism. And we're able to support this with subsequent studies that we did to confirm that, looking at assemblies and the location of this in the actual genome sequence. And that's concerning because if that jumps into the chromosome and translocates there, there's a risk that could become a fixed feature of that particular cluster of sequences. So it's important for surveillance reasons to understand that. So just to sum up, I hope I've been able to convince you of the utility of including phylogenomic and phylogenetic analyses in a genetic epidemiology study, as well as other types of data. So in this particular example that we've walked through, we've included clinical information on cases and carriers. We've included temporal data that included the date of isolation. We've included country of origin data. We've included genome-derived data on genotypy genotypes, antimicrobial genes and mutations that cause resistance, and also plasmid marker genes to help us understand some of those dynamics. So from this data, we've identified local transmission dynamics. We've identified occasions of regional spread, as well as global transmission dynamics from different regions of the world to other regions. We've identified emerging AMR in the form of those ciprofloxacin non-susceptibility mutations I pointed out, and changing dynamics in terms of chromosomal integration that's occurring during the study period of that integration of multi-drug resistance into the chromosome. And all these data are informative for control and intervention studies. And if you'd like to find out more about this work, I do encourage you to take a look at the publication from Sam and his team in eLive. So I hope you've enjoyed today's webinar tutorial, and I'd be very happy to take any questions that you might have.
All right, thanks everyone. We will now open the floor for Q&A. Please feel free to type your questions either in the Q&A bar or within the webinar chat. I see there's a question by Tracy Colbert Joshua. She says, I noticed that on the slides showing the 3 lineages, some branches are quite long. When should we be concerned about long branches and when are they valid?
Yeah, that's a fantastic question, Tracy. So with some phylogenetic methods, they all have their benefits and drawbacks. So some people might be concerned, say, about long branch attraction. That is where divergent sequences are mistakenly clustered together by the software. So I think this really comes down to good QC a lot of the time, or quality control. So I would really be considering, am I sure that the organisms that I put into my phylogenetic inference software are indeed, in this case, all typhoid? Could there be a chance that there is something more distantly related in there? Am I certain that they are pure sequences of a given organism? It's not uncommon to see some read leak creeping through in some sequencing runs, or indeed sometimes mixed samples of multiple genotypes of a given pathogen. So it's really, if you've done all of those things, and you've looked at the differences in substitutions between your isolates, then there are some other things you can investigate. For example, transmissions and transversions that might elongate your branches. But I think the first port of call should always be really good QC before you get to the stage of inferring your phylogenetic tree. So I hope that helps.
All right. We then have another question from an anonymous attendee. They say that, sorry, the question is, how do you determine an appropriate reference for a rooted phylo tree?
That's a really great question. So it really comes down to a lot of the time what you know about the pathogen and community consensus. So if I'm working with data from a new pathogen, firstly, I want to know all about it. So I'm looking at all the different publications that have focused on that given pathogen. And usually it does emerge that there are popular outgroups or popular reference sequences. I'm not sure which particular one you're asking about in that case, that the community is using. And unless I had a good reason to use something different, I would probably stick with what they recommend. But broadly speaking, speaking from my own experience, which is largely on Salmonella, I would be looking to use a different serovar of Salmonella enterica subspecies enterica, or indeed a group of those. But I wouldn't go so far as to use something like E. coli that's going to be quite distant from that. I hope that helps.
Right. Thank you very much. There is another question by Rudan Ali. How to choose which algorithm to use? Does a phylogenetic tree constructed using nucleotides superior than one using amino acids?
Yeah, another really great question. So I think it really depends on what your goals are for your analysis. Certainly, if I'm looking for transmission of a pathogen from one location to another and I'm working with bacterial data, what I really want to do is to infer a core genome alignment of that particular pathogen. And then I usually want to be doing that using a SNP-based method because I want to be capturing not just genetic change within genes, but I want to be capturing genetic change between genes that might also be informative for understanding the evolutionary history of the particular pathogen. And the method that you could choose, you've got lots of different options. There's distance-based methods like your neighbor joining that are quite fast. You've got more robust methods that have a lot more certainty to them, like maximum likelihood, that are more computational intensive. And you've got your Bayesian methods where you want to really be looking at time and want to be putting dates to when different variants of concern have emerged in a population. So it really depends on what your goals are for your study. In particular, I would consider doing amino acid trees, just thinking back to your original question, where I wanted to know specific information about maybe a family of resistance genes. That might be a use case where I would consider using amino acid sequences. I hope that helps.
All right. We then have a question in chat. Kind of indicate the approach of beast approach analysis. Did you use mapping reads to the reference, or did you perform core alignment SNP analysis before performing beast analysis?
Really great question. So I think this might be the focus of a future webinar, so I didn't want to overlap too much. But the process that we went through was indeed mapping to a reference sequence. And for typhi, we tend to use CT18, where we're making sort of more global trees with lots of different lineages in them. But we tend to also use it for single genotypes too, because there aren't that many other reference genomes around in public databases, but also it's very well annotated. It was manually curated by colleagues at the Sanger in 2001. We have a lot of information about what all the genes do, and it was primer walked and finished to high quality. So we do tend to use that. So we mapped to that given reference. We then called variants, and we used a mapping pipeline called RedDog to do all that. We then made core genome SNP alignments. And I believe the threshold we used for that was 95% conservation. It's in the paper, but I'm pretty sure it was 95. We then fed that through to gubbins, first converting it back to a whole genome sequence alignment to give the spatial context to it. We then ran gubbins to filter out any recombinant regions that were present in there. And we also masked out prophage regions and repetitive regions where we couldn't confidently call SNPs before we did that. And then once we had that recombination filter alignment, we inferred a maximum likelihood tree using RAXML. I hope that helps.
Right. Then we have another question by anonymous attendee. How can SNP matrices be utilized during outbreak investigations?
That's a really great question. So what it can help you do is put a number to how similar or dissimilar a cluster of sequences are. So if I was looking at a relationship where something had moved from India to Nepal, there's a famous triple mutant typhoid that's done that. I can actually put, a number to that to say they are quite closely clustered together. And the number that you might want to use really depends on how quickly you expect that pathogen does evolve. For example, Salmonella typhi, it's quite slow. If I'm talking about recent transmission, I would expect to see 0 or no SNPs different for something quite recent. But if I'm allowing more time for that to happen, it could be my divergent, that number of mutations would go up. So it's context dependent on the type of events you're trying to capture in your analysis, but also how quickly that pathogen evolves. So I hope that helps. In the previous example with Ugandan and Kenyan sister strains of EA3, they were 13 SNPs apart, and we just put that number there to show that they are reasonably closely related to each other. I hope that helps.
Right. Just due to time constraints, we're going to be taking 1 or 2 more questions. We have somebody who has asked, again, another anonymous attendee. They were wondering specifically how to incorporate specimen data to calibrate the molecular clock/phylogenetic distance on the x-axis. Was there any particular software that was used?
That's another great question. So that particular analysis was done using BEAST. There is BEAST and BEAST2. They aren't a predecessor and a new one. They're 2 software tools that are similar but developed concurrently. That has really been historically the workhorse of dated phylogenetic analysis. Other tools that are really great that are newly emerging that can do that analysis on a greater scale might include backdating. That's a tool developed by Zoe A. There's an R package that's available. There's also tree data that you might look at as well. So those tools are definitely great if you've got large sample numbers and you want to do a large scale analysis. I hope that helps.
All right. Then we have another one related to software, but which tool was it that you had used to edit the tree? For example, the last one that you presented on.
To infer the tree?
So which tool did you use to actually display the tree and edit it accordingly?
There's a wonderful R package. Yes, sorry, I didn't mean to speak over you. There's a wonderful R package out there called ggtree. There's a pretty good documentation on it as well. It's a wonderful R package, and I highly recommend it to people. If you're looking for something else that you might like to use, there's also ITOL out there that a lot of people find really helpful, and microreact is also another good one. But I quite like ggtree.
All right. Well, then just do one last question. So this is by Curtis. What are your thoughts about using a sample and its de novo assembly that's part of your set of samples as the reference sequence for creating core genome alignments and building trees?
Yeah, so this is done quite a lot. It's a really, really good method. I think this might be the discussion of a future webinar. So again, I didn't want to touch on this too much. But broadly speaking, it's a great approach. Where I would favor that approach over the mapping SNP calling approach we've already discussed in the Q&A would be where I'm expecting that a sequence is a particular pathogen is more divergent. So this might be an approach I would consider for E. coli, for Klebsiella, where I'm looking at sort of quite a broad range of different genotypes or sequence types or lineages. The caveat to it being that while it is a great approach for those types of organisms, you do encounter some data loss. So with those methods, you'll tend to be just looking at variation in genes, missing mutations that might be informative between genes. So I think that's a consideration, but it's an excellent approach as well.
I see, sorry, just one last question that was posted.
Of course.
Here. How do you see the role of pathogen watch for these epidemiological investigations?
I think Pathogen Watch is great. I use it myself when I want to do something quickly or I want to first look, but there's people writing papers here, full genetic epidemiology studies that have utilized Pathogen Watch, and it's a fantastic platform. Our genotyping scheme is embedded within that, so you can have all the functionality of genotypy, AMR libraries that are customized to Pathogen Watch, And the benefit of this is that all of the global data that was recently curated by the Global Typhoid Genomics Consortium, with which I'm a coordinator, is available in there. So you immediately have this ability to generate rich global trees using more than 13,000 genomes to contextualize your data and understand the epidemiology with having to bring all that data onto your own cluster. So I think it's great, and it eliminates a lot of need for, compute overheads that some cases are difficult for us to obtain and maintain. So I love Pathogen Watch. I think it's fantastic.
All right, excellent. All right, then. Thank you very much, Dr. Dyson, for your insightful presentation on the introduction of phylogenetics into public health, and specifically advancing our understanding of pathogens and how to guide better treatment strategies in the face of antimicrobial resistance. We look forward to any contributions from the community. Thank you very much for your attendance towards the phylogenetics working group and the upcoming training. We do believe that you guys will be an extremely valuable resource for everyone working on the intersection of phylogenetics and public health. I just, again, like to thank everyone for joining us today and encourage everyone to engage with these really exciting initiatives. And please feel free to reach out to any of our organizations or members regarding further discussions or collaborations. Thank you again, Dr. Dyson, and thank you everyone for your attendance.
Thank you for having me.